Biomarker has pioneered the development of genetic technologies and industry since 2009 and owns more than 60 invention patents and over 200 software copyrights. Additionally, Biomarker has contributed to over 1,000 scientific programs published in high-level academic journals with a cumulative impact factor of more than 4,500. This continuous innovation in scientific research fuels Biomarker to become one of the premier promoters of genetic technologies in the era of biotechnology. Biomarker aspires to simplify gene decoding and accelerate advances in scientific research and is committed to provide standardized, customized and comprehensive technical services and solutions to customers all over the world. We have established 4 business segments devoted to high-throughput sequencing, bioinformatics, BMKCloud, mass spectrometry, and intelligent manufacturing. In the field of next-generation sequencing, Biomarker leads functional genomics with SLAF-seq. SLAF-seq has accurately identified hundreds of vital functional genes that closely relate to human lives and has served a role in over 30,000 projects. Since third-generation sequencing came onto the stage, Biomarker became one of the earliest players in this new arena. We are proud to have introduced the PacBio platform-based tandem full-length 16S, 18S, ITS, amplicon sequencing technology and have launched Nanopore platform-based full-length transcriptome sequencing services, which brings microbiome and transcriptome study to a new level. The internet era has provided great opportunities in genetic technologies. Biomarker has efficiently and professionally integrated multi-omics-based big data and an online interactive analysis platform and high-performance cloud computers on BMKCloud, a self-developed online biocloud system. The platform is designed to help users interpret biodata and promote scientific research and health management, leading the generation of cloud-based genomics and biodata. Guided by the core values of continuous innovation and the pursuit of excellence, Biomarker provides its customers with high-quality services and products. At the same time, Biomarker has built state-of-the-art and automatic professional molecular experiment platforms, automatic and intelligent bioinformatic analysis platforms, and an information-based and standardized system to manage production, sales, staff, and finance. Biomarker will continue to create greater value for its customers and lead the industrial transformation with high-quality innovations to realize its dream of benefiting humanity with genetic technology. Biomarker Technologies. Innovation leads to wonders.
Everyone, the webinar will start in 5 minutes.
Biomarker has pioneered the development of genetic technologies and industry since 2009 and owns more than 60 invention patents and over 200 software copyrights. Additionally, Biomarker has contributed to over 1,000 scientific programs published in high-level academic journals with a cumulative impact factor of more than 4,500. This continuous innovation in scientific research fuels Biomarker to become one of the premier promoters of genetic technologies in the era of biotechnology. Biomarker aspires to simplify gene decoding and accelerate advances in scientific research and is committed to provide standardized, customized and comprehensive technical services and solutions to customers all over the world. We have established 4 business segments devoted to high-throughput sequencing, bioinformatics, BMKCloud, mass spectrometry, and intelligent manufacturing. In the field of next-generation sequencing, Biomarker leads functional genomics with SLAF-seq. SLAF-seq has accurately identified hundreds of vital functional genes that closely relate to human lives and has served a role in over 30,000 projects. Since third-generation sequencing came onto the stage, Biomarker became one of the earliest players in this new arena. We are proud to have introduced the PacBio platform-based tandem full-length 16S, 18S, ITS, amplicon sequencing technology and have launched Nanopore platform-based full-length transcriptome sequencing services, which brings microbiome and transcriptome study to a new level. The internet era has provided great opportunities in genetic technologies. Biomarker has efficiently and professionally integrated multi-omics-based big data and an online interactive analysis platform and high-performance cloud computers on BMKCloud, a self-developed online biocloud system. The platform is designed to help users interpret biodata and promote scientific research and health management, leading the generation of cloud-based genomics and biodata. Guided by the core values of continuous innovation and the pursuit of excellence, Biomarker provides its customers with high-quality services and products. At the same time, Biomarker has built state-of-the-art and automatic professional molecular experiment platforms, automatic and intelligent bioinformatic analysis platforms, and an information-based and standardized system to manage production, sales, staff, and finance. Biomarker will continue to create greater value for its customers and lead the industrial transformation with high-quality innovations to realize its dream of benefiting humanity with genetic technology. Biomarker Technologies. Innovation leads to wonders.
Good morning or afternoon. This is Lydia from BMKGENE. I'm thrilled to welcome each one of you to our latest webinar. Today, we will explore from data to report in BMKCloud in the case of mRNAseq with reference genome analysis. Our webinar today will last for about 35 minutes, followed by a dedicated 5 minutes for your questions and answers. You can freely submit your questions by using the Q&A button at the bottom of your screen. We are looking forward to receiving your inquiries. And if any questions are left unanswered, we will contact you to reply via email. Also, we'll share the slides and the recording of this webinar via email. In this webinar, we will explore the application of mRNA sequencing on BMKCloud, a comprehensive bioinformatics platform. The following topics will be covered. Firstly, introduction to BMKCloud. Discover the powerful bioinformatics platform offering a range of tools, applications, and data mining services tailored for microbial research. Secondly, analysis workflow from data to report. A deep dive into the main findings and insights generated with BMKCloud's amplicon analysis workflows. Thirdly, update final report. We will explore situations of results need to be updated. Update report, remove samples, change parameters, and update groups. Fourthly, advanced analysis tools. In-depth data filter by 5 minutes in 3 situations. And drawing tools. One-click drawing, Venn diagram, and customize, and advanced analysis, WGCNA, and cluster. Before we start, let me introduce our distinguished speaker for today's webinar, Johnson. Equipped with extensive expertise in bioinformatics analysis, shines in his role. Holding a master's degree from Beijing Forestry University, China, he embarked on his professional journey as a bioinformatics analyst post-graduation, specializing in high-throughput sequencing data analysis, especially Pacific biosciences data. Johnson honed his skills with precision and dedication. His career trajectory led him to BMKGENE, where he transitioned to the overseas bioinformatics department. Here, he assumed the pivotal role of cloud platform product manager, steering the team towards innovation and efficiency. With a blend of technical prowess and strategic vision, Johnson continues to make significant contributions to the field, driving advancements in bioinformatics with unwavering determination. So without any delay, let me hand over this virtual stage to our master, Johnson.
Thank you. Hi, it's Johnson. Today, I will introduce to you how to use BMKCloud to easily and quickly from data to report finish an analysis using an mRNAseq with reference analysis in a case. This presentation mainly includes the following 5 parts. First one, BMKCloud introduction. I will introduce you about BMKCloud as a bioinformatics platform service. And number 2, analysis workflow. We will truly using BMKCloud from data to a final analysis report. And the third one, I will demonstrate to you about how to update final report, such as remove samples, change parameters, and change group settings. And fourth part, I will introduce you about how to use BMKCloud doing advanced analysis, such as data mining, one-click drawing, and summarize of today's webinar. First of all, you should know that BMKCloud can offer a one-stop solution. We have bioinformatics tools, such as image generator, sequence analysis tools, and data processor. You can use our online analysis applications to do some basic data analysis. BMKCloud provides various analysis applications. After you got the final report, you can do data mining or customize images without limitation. You can change colors, reset groups, or other customized setting by yourself. So, how does BMKCloud provide service? Let's start from data. There are 3 situations. Number 1, sequencing and analysis completed in-house. And number 2, only sequencing conducted in-house without bioinformatics analysis. And number 3, users with their own data. For data sequencing in-house, we can push data or report into your BMKCloud account. You can try function online by yourself. For the third part of data for yourself, we will recommend opting for data analysis on BMKCloud to avoid the significant time consumption of data transmission and the instability caused by the network fluctuation. Let's recognize the BMKCloud interface. This is the screenshot of main interface of BMKCloud. We will see the same page after you log in into our platform. Using navigation bar on the top, you can manage your account, change language, and connect us by email. But actually, you can connect our cell managers. Next, you can see your account main information, including your storage space left or analysis limitation. Navigation bar on the left is very powerful. You can find almost every function of BMKCloud here. You can manage your project, such as upload or delete data, monitor your analysis task here. You can also find analysis applications of BMKCloud in bioinformatics, and I will introduce to you this part in detail later. Let's leave this aside for this moment. In this part, I will tell you about task management. If one wants to complete an analysis, we should prepare data, do analysis, download the results. In BMKCloud, especially sequencing in-house, you can simply divide whole pipeline into 3 parts, including task submission, task monitor, and task download. Now, I will demonstrate to you how to about mRNAseq reference analysis using BMKCloud step by step, ensuring that you can finish mRNAseq analysis by your own. Let's turn to BMKCloud homepage. This is the main interface of BMKCloud, and I just introduced the elements to you of this page. In case we want to finish an mRNAseq reference analysis, turn to the left navigation bar and find this bioinformatics applications. Click it. And here is all of the analysis applications we can use on BMKCloud. And today we will use mRNA reference in a case. So click it. Open the application. Here is the main interface of task submission. I will introduce to you about the parameters. On the top project name, you can change the project name as some strings as you can remember. For example, I want to set the parameters as today's data. It's October the 15th. And here, step 1, the raw data loading. We can loading the raw data included in our BMKCloud account already. Or we can push your data sequencing in BMKGENE into your account, and so you can select the data sequencing in-house. Here, I use the data I pre-prepared. So here, I have 9 samples of some plants. In this box, you can change the sample's name. Our cloud platform will automatically pair the parent reads, such as here, the 408 reads 1 and 408 reads 2 will automatically paired. And you can change the name. Maybe I want to change this sample into the A1 repeat 1. And the next sample is A1 group repeat 2. And the third sample is the A1 repeat 3. And this sample name will displayed in your final report instead of this long string. This is not recognizable, but this you can divide the samples. After you load the raw data, you can turn to the next step for the main parameter settings. If you don't know how to change the this analysis workflow version, you can set this by default and the lib type. Most of us is for unstringed the lib type. If you have the string type, you can change it here. But for most of us, we can use this. And the third one, the reference genome. For mRNAseq reference analysis, the most the reference genome selection is the most important part of the task submission. So we can select the reference genome species. For the species, we can just click type into some letters. The system will automatically pick the species out. And after you select the species, all the reference genome version available will listed below. You can choose one available reference genome already on BMKCloud, or you can connect us for some version we don't have. If you don't know how to choose the reference genome, you can look at here. For the last line, the data update type. For most of the analysis, you can change the most recent one. For this is I prefer maybe I choose this tier 10. This is the most published and accessible version. Maybe I can use this for the further analysis. And third one, differential expression analysis. This part is the most focused on part on mRNAseq analysis. And you can change the mathematics parameters. For most of us, we have reputations for the samples. So we can group the samples into, we can set the samples into groups here. Do you need to group? Most of us need the samples into groups. Maybe I have the first group named A1, has the sample A1, A2, and A3. This is the first part, and add group. I want to set the second group, maybe it's named B1 group, has the samples M4, M5, and M6. And add the third group, maybe it's named C1. This group has maintained the samples of M7, M8, and M9. Here I have the three group settings, and I want to set the compare settings for the control group and the treatment group. Maybe the C1 group is the naive group, so I set this as the control group and the B1 group as the treatment group and add. So this compare groups settings will analyze in the workflow. And next part for the C1 group compare with the A1 group and add. So I set two groups of analysis. Let's back to here. This is the mathematics parameters such as FDR and fault change or the automatically parameters adjustment. If you don't know how to change these parameters, you can leave this by default and turn on automatic parameter adjustment button. On our workflow, we will automatically change the parameters for your analysis to avoid the less differential expression genes number, all the out of group settings. After you setting all these four parts, you can just submit the analysis here. Click the submit. Oh, project name. Click the submit, and the analysis with the parameter we just setting will automatically upload to the BMKCloud. And the BMKCloud and the platform will automatically finish the analysis workflow. You can just download the report after the workflow is finished. So talk about workflow after we just submit the analysis. Where can we monitor the analysis? Let's back to the homepage of BMKCloud. On the left navigation bar, we have here project, tasks, Here you can see the task we just submit. Okay. We can have the project's name for the today's data. This is the project we just submit. And the label here is running, which means the report is the analysis is still running. You can monitor your task here. If the analysis is finished, the label here will turn to the complete, and you can click into the final report and do the further analysis. For the task, for the report download, I have a pre-prepared analysis report here, and let's turn to this. Here, this is the mRNAseq reference analysis report I just pre-prepared, and if you can get the same report, if you submit after you submit the analysis on your own. You can check the HTML report in this page, and you can download the report into your personal computer on the top. Here, I can click the download button, and in the opening window, you can choose which format you want to download, such as HTML format or the PDF format, or just this is the you can download the report in this two font without the specific files in detail, or you can download all results. Click this, you can download the zip file containing all the results, such as the FASTA file, GFF file, or the other bad files. This contains all the files. So this is how you can use BMKCloud submission, monitor, and download the report. Let's back to my slide. Let's summarize here. We talked about submit analysis using BMKCloud. Choose mRNAseq reference you need. Select the data in BMKCloud and set analysis parameters. We talked about how to monitor tasks, how to tell whether process is running or finished, and manage all your tasks. After finish analysis task, you can then download final report into your personal computer. You can choose any format you need: HTML, PDF, or total zip file. But after we got the final report, we may meet these situations. On the left, you can see of the evolution tree. There is a symbol out of this big group. I want to remove these symbols. On the right, the differential expression genes number statement plot. And we can see there are too many differential expression genes. This is hard for our further analysis. For most of the situation, we want less differential expression genes for the further analysis. We can do the experimental procedures or for others. So how to deal with this situation? Maybe I want to change the p-value or the FDR or the full change, may change this. So after we got the final report, how to solve these situations? Let's turn to BMKCloud and I will show you step by step. Okay, let's back here. This is the report I just showed you. I didn't recognize that on the left is also the navigation bar. This is the advanced analysis or the update the final report buttons here. Just we talked about I want to change the sample settings and the group settings. And I have too many differential expression genes, and I want to change the mathematics parameters. So the first one, I want to remove some samples. How to do this? First, I have a new report name. I set this into today's data. And I want to remove H3R1 sample. So I have the report name for me. You can just change this anything you want. The first step, I want to remove H3R1 samples. So I click all the samples here. If you click all, you will update group with all the samples. If I want to remove H3R1 samples, just unclick it. This sample will not display in the updated report. And there's and by this way, you just remove a specific sample. Maybe you want to remove more samples, such as I don't like the naive one and the H3R2. This is all depend on you. I just want to remove the D3R1 or H3R1 sample. So I unclick the H3R1 sample and click other samples. The next step, I want to change the differential expression analysis parameters. Maybe I have too many differential expression genes, so I want to make the fold change bigger. Here, the default fold change is 1.5. I want to change it into 2. So I will get regularly I will get less differential expression genes number. And the third part, you can change the group settings here. Maybe I have the wrong group settings, or I want to remove a sample out of some groups, or combine two groups into one group. So how to do this? In my previous analysis, I have D3 samples in D3 group, H3 samples in H3 group, and naive group. I have 3 groups. I want to combine D3 group and H3 group into one group as treatment. So I can here. This is the same function in the task submission. I want to has the treatment group has all this. Okay, then I could combine the 5 samples into one group named the treatment, and I add a naive group as the naive repeat 1, repeat 2, and repeat 3. So I update the group settings. I have 2 groups here, and change control group is naive, treatment group is treatment, and add. After this, I change the sample settings, change the analysis parameters, and change the group settings. Submit the task. BMKCloud will automatically update your final report and finish this with the change we just submit. How to monitor the update workflow using BMKCloud? We just submit an analysis, update the final report back to the homepage of BMKCloud and the same page as project tasks. Here you can see this project we just submit. It's based on the mRNAseq reference Johnson. I just prepared and it's still running. If the label turns to complete, the analysis will the analysis is finished. You can check the update report. But do you know how to check the final updated group? Where can I find it? So my updated group report is based on the previous group report here. This is the report before I remove the samples and change the group settings. And here, see the button, update the final report, new report, click it. This here, you can see all the report we just update. This 2 report is the I just prepared. Click it, and you can turn to the report you just update in a new window. So let's back to my slide. Let's summarize here. We just talk about how to adjust groups and delete samples. and how to modify the parameters you just submit and update, finalize the report. You can submit it, monitor it, and download it. In the final report, we will obtain many DEGs. It depends on analysis parameters, but even a very suitable parameter setting, we will get many DEGs. Usually, we only need a few genes for further analysis, sometimes even one, particularly some functional genes. So, what we should do to filter gene set? Let's think about 3 situations here. First, your research group is studying topics related to sugar. After obtain the final report, you want to first retrieve genes related to sugar pathway to check their expression levels. You want to select genes with functional annotation results, such as the GO and KGG pathway, or NT and NR database. In this situation, you can filter genes by feature nodes. Second, you already have the IDs of several key genes, maybe from a published paper or molecular expression experiment, and want to check the expression levels of these genes. So you can easily filter genes by gene IDs or gene symbols. And the third one, use research request finding genes with specific sequence feature, such as containing a particular motif. So how to deal with these situations in BMKCloud? I will demonstrate to you step by step. Let's turn back to the report page of the updated report. This is the main interface, and you can see on the left is the navigation bar. In the middle is the main report in HTML format. We want to filter gene set, or in other words, I want to select some genes. So click here, gene expression, gene search. This is the function that we can change. We can select the gene set or select the specific genes. We have file method for the gene search. We can search gene with the first feature nodes such as GO and KGG, and some functional annotation results. This is the most useful part. And you can use the gene ID, gene name, or sequence information as motif or the peaks. I will demonstrate you in feature nodes, such as I just talked about. We want to search genes related to sugar. So just type the sugar keywords into this box. Here, I have 28,000 genes in total. And after we search that, this may take a while. This depends on how many genes do you have. And we got 47 genes in total. These genes are related to sugar. But I want to notice you that the current research result is based on the previous result or the result we just selected. Maybe I want to search the genes about ATP. Here, notice that I searched the ATP based on this feature nodes. This feature nodes is all the genes about sugars. Just click the search. See, I have a total 19 genes related to sugar and related to ATPs. And back to the whole gene set, we have 28,000 genes. What if we want to search ATP here? Just click it. And we got 2,600 genes in total. This is not same with the feature nodes result 3, because feature nodes result is based on the result previous, feature nodes result true, but the feature nodes result 4 is based on the group set, because a whole gene set. So this is the difference. I want to show you another example. Click the gene search in a new window. This is the whole gene set. I will demonstrate this part again. First, maybe I want to search the gene about related to ATP first. So I have 228,000 genes in total, and search genes related to ATP. Click search. And I have the 2,600 genes in total. I want to search the gene related to sugar based on this feature result. So just click this feature result and type into the sugar. I have 90 genes in total. And what if I want to search the gene? I want genes related to sugar, not based on the ATP. Just click the gene search result 1. This is the whole gene set, and search sugar. You can see I have 407 genes in total. This is the first part about how to search genes. And I want to show you about how to do a one-click drawing in BMKCloud. After we search the gene set, we can just save the list in Excel format, or just analysis result for the whole tables. But this is not a visualization result, and it's not very rectangle. So BMKCloud have a very convenient part for the one-click drawing. This one-click drawing is based on the gene set we just obtained here. Maybe I here use the feature nodes result 4. This is the gene related to sugar in whole gene set. And I use this 403 genes doing the one-click drawing. After we clicked it on the left, all of these images will automatically finish taking a while. Maybe because of the numbers of the genes, it may take a while. The background of these features is still white, and here you can see this. The background of COG classification turned to gray, which means this image is finished. Click it, and you can see the image using the data of this gene set, this 403 genes. Click the image, and you can see the more functional settings. On the top, you can change the parameters and customize the settings, such as text adjustment. You can change the font size, font family, or the font color here. I will show you a special way. I want to change the font into red. So you can see all the font is turned to red in this image. And you can also change the color settings, because maybe I don't like the color in the bar plot. You can change it here. Layer setting for the labels of this part, and the personalizations. You can try this part. This is not the same in the different images, and you can try this part on your own. We will talk about this in details on next webinar. Here, I want to show you that you can intersect the gene sets using the image. Maybe I want this G group. G group is the target gene set. I want to do the further analysis. So just click it on the Here you can see the data filtering. Click it, and you can get the gene set containing the 148 genes in total. This genes is based on the gene feature notes about sugar and the G group in COG classification. It's very convenient. You can just select the gene set in the image. You can try all these advanced analysis tools in BMKCloud, or you can come into our next webinar. We will talk about this function in detail. So let's back to my slide. In our real study, target gene selection is a complicated work. We must track differential parameters, so there might be many middle results. If we save all these middle results in our personal computer, it's very hard to arrange the files. But you can easily arrange these results on BMKCloud. You don't have to save all the middle results, just the final gene list. I think it's very more convenient, much more convenient in our daily research. And we just talk about how to do a one-click drawing. It's easy to understand. Get your gene set, do one click, and wait for the result. Here are some notifications. The green background means that the image is done, while the other type with blue title means the working is still running. We must keep in mind that visualization results are based on the current gene set. In this screenshot, this means all the images are based on the 403 gene above. Let's summarize here. We have 4 topics today. We just talked. The first one, I just introduced to you about BMKCloud and how BMKCloud platform provides service in different ways. And number two, analysis workflow. How can we just using BMKCloud from data to a final report? And number 3, we talk about how to update the final report. We can change the samples, change the mathematics parameters, and change the group settings. And the last part, we talk about advanced analysis. We can get the gene set filtering and do the one-click drawing. Last webinar will presentation in the next week. We will analyze real data and talk about advanced analysis tools in details. That's all. Thank you. Any questions?
Okay, thanks for Johnson's presentation. He explains in great detail. I believe that we have a good understanding of from data to report in BMKCloud in the case of mRNAseq with reference genome analysis. Now we have received several questions regarding this. The first question, can I update report again if I already changed parameter and get updated report? Let's ask Johnson to answer for us.
Oh, this question. We just demonstrate that we can update the final report using BMKCloud. But after you got the updated graph, you can see the update the final report button also in the navigation bar, which means you can update the group final report again and again until the result suits your design well. You can update the final report without any limitation.
Okay, another question. Is there any explanation in detail for advanced analysis tools usage?
Oh, advanced analysis tools is a powerful part of BMKCloud. The first one you can, let's back to the BMKCloud and I will show you that here. You can see on the left is the advanced analysis tools, and you can see this label, which means this analysis tools has a user guide video on YouTube, and you can watch this. You can watch this on yourself, or you can just come into our next webinar. We will talk about this part in detail, and you can ask me any questions if you have.
Okay, thanks for Jonathan's answer. So as we conclude today's session, I want to express our gratitude for your participation. And later we will send you our email and the webinar summary and the promised recording, and make sure you check our social media page for future webinars. Thank you again. Have a nice day.